Hello, everyone. Thanks for coming to my talk. I really hope you like it. So let's get started. So I began working with Cladoniaceae from monitoring two populations that occur in the Caribbean island of Puerto Rico. Populations of these species are distributed in a lower elevation site near the coast here, marked in yellow, and other populations that are high up in the mountains of Maricao, here marked in green. So one of the things that I was really interested early on with this species was to try to see if these populations were either part of the same species or perhaps populations of different species. So I carried out a simple pilot study looking at single locus data and phylogenetic analysis to try to go around this. And this analysis was inconclusive. Some of the analysis showed that suggested this belonged to the same population and thus the same species, and perhaps, and the other analysis, sorry, looking at other genes, I should say, suggested that maybe these were separate populations and perhaps separate species. So the other thing that came from this preliminary studies was that Cladoniaceae was consistently found nested within another well-known species called Cladonia subtenuis. Cladonia subtenuis is morphologically very similar to Cladoniaceae, with the difference that one produces usnic acid and the other one doesn't. So that's what we use to separate the species. However, sandstedei produces atronormin, which is also used for taxonomic purposes too. The other thing that makes it a little bit more complicated is that these two species have some degree of overlap in their range, especially in the southeastern US and in the Caribbean, where both species are found. So to study this in further detail, I decided to implement a next generation sequencing approach, specifically to evaluate first and clarify the evolutionary relationships and phylogeography of these species, the usefulness of secondary chemistry for discriminating between them. Also interested in assessing genetic differentiation associated with geography, and perhaps discuss potential taxonomic consequences. So for the methods, I perform regional sampling. Specifically, I sample populations in southeastern US, here marked in gray, and from populations in Cuba, marked in purple, and from Jamaica, marked in blue, and of course, the two main populations in Puerto Rico, which are marked here in green and in yellow. I identify individuals based on broad morphology, of course, but mostly on secondary chemistry, which was based on TLC analysis. For the genetic analysis, I generated RADseq data. RADseq is a method that allows us to obtain sequences from flanking regions associated to restriction sites in the genome of many organisms. So I use bioinformatic pipelines, stacks, and iPyrat to process these data sets and obtain alignments and SNP data files that I use for the analysis. And for this analysis, I did a phylogenetic analysis with RADseq-ML. And for visualizing population structure, I used discriminant analysis of principal components. This is a non-parametric method that uses a PCA in combination with discriminant analysis to visualize and separate individuals in discriminant function space. Specifically for this analysis, I perform a de-novo clustering using a k-means clustering algorithm. This is the main tree from this analysis. Strongly supported clades are shown with the black and gray circles. And I'm also coding each individual using different colors to code the presence of substances in each of the individuals analyzed. And then to the right, we have the geographic coding. And the first thing to know, and I want you to focus, is in the continental clade. The continental clade has a strong support at the base, but as you can see, there's no major phylogenetic pattern emerging from all these individuals, and most importantly, no clear correlation of chemistry with topology or phylogenetic patterns. However, when we look at the Caribbean, we note a different scenario where each island, individuals of each island came out as strongly supported and very divergent clades associated to each of these islands. Especially important to note is Jamaica. I have individuals of both sandstedei and subtenuis within this island, and I found that the individuals were more closely related to each other than they were to the rest of the individuals in the whole sampling. Also note that all the sandstedei individuals from Cuba and the populations in Puerto Rico, the chemistry was consistent, which contrasts with what we found with the continental level. This is the results for the DAPC. All the individuals are the dots here and discriminant function space. And at the bottom, we have a structure like a plot with the degree of membership indicated by each of these columns. And first to the left, we need to highlight the continental level data. The method inferred two main populations with really poor separation between them. Important to note here is that all the individuals from Jamaica were inferred to belong to one of these continental populations, suggesting they might belong to the same species. To the far right, we have the individuals from populations in Puerto Rico and from Cuba. So all individuals from Cuba were found nested within the population of Maricao. There's a lot of scatter in this side of the plot, which indicates genetic variation of all these individuals. However, it's also indicative that there might be some degree of divergence occurring here. So to conclude, secondary chemistry, we believe it's poor for species discrimination in this group, particularly at the continental level. This shouldn't surprise us, as has been indicated for other Cladonia species complexes. We found some degree of conflict with the Jamaican population specifically. One method making it closer to the continent and the phylogeny, separating it from the rest. However, Cuba and Puerto Rico were consistently separated in both analyses, and this might deserve taxonomic recognition. Further sampling in the Caribbean and southeastern US is needed to really resolve the taxonomic issues in this. We really couldn't obtain samples from the southern tip of Florida and from Hispaniola, which both have populations apparently of both species. So we will need further data on these areas to clarify this. Also to remind that all the populations from Maricao and Vega Baja at this point are hard to separate, and they as of now should be considered part of the same species. However, there might be a process of incipient speciation going on that needs further analysis. So with that, I want to say thanks to the organizing committee for the AIL, to my colleagues Sean and Malcolm, Robert, Viviana, Roger for helping with access to field sites, permits, samples, and to my funding sources, which I am recognizing in these images here. And of course, to all of you for hearing me out. I'm super happy to take questions. Have a good one.